In our final lesson on nitrogen metabolism, let's examine catabolism of pyrimidines and then see how ribonucleotides are converted to their deoxy forms. As in purine catabolism, pyrimidine nucleotides are first broken down to the nucleoside and then hydrolyzed to release the nitrogenous base. Since pyrimidine bases have a simpler ring structure than that of purines, the ring can be opened to form a beta-amine product, and that can feed into other pathways. This means that there is no real pyrimidine waste product. In all organisms, the synthesis of deoxyribonucleotide diphosphates is accomplished by reducing the ribonucleoside form, as shown below. Note that since a CO bond has been replaced by a CH bond, the carbon has been reduced. This reaction is catalyzed by ribonucleotide reductase, and thioredoxin is the direct reducing agent, as outlined below. First, thioredoxin is reduced by a separate enzyme, thioredoxin reductase, by the process outlined on the left of the figure. Electrons extracted from NADPH are first passed to an FAD cofactor and then to an oxidized disulfide bond. Ribonucleotide reductase then uses the electrons in the reduced sulfhydryls of thioredoxin to convert the nucleotide to its deoxy form. The products include deoxy-UDP, CDP, GDP, and ADP. Note, however, that deoxy-UDP is not found in DNA. Hydrolysis of DUDP gives DUMP, which is the substrate for the reaction catalyzed by thymidylate synthetase. It uses the one carbon carrier THF to methylate deoxy-UMP to deoxy-TMP, as shown below. Nucleotide biosynthesis is of course active in all rapidly growing cells, including cancer cells. Thymidylate synthetase is therefore a common target in chemotherapeutic agents. Dihydrofolate is another of their targets. The effect of these agents is that cancer cells are not able to generate the nucleotides needed for DNA synthesis, and that makes these drugs lethal to those cells. The consequence, though, is that the drugs are also lethal to other rapidly growing cells, including normal, healthy ones. This is why there are certain side effects to these drugs related to the death of rapidly growing normal cells like hair follicles, intestinal epithelial cells, and immune cells. These facts but illustrate how an understanding of cellular metabolism helps to develop methods of treatment for diseases and disorders related to that metabolism.